Hemophilia is a genetic disease where even a small bruise can become a life threatening medical event. Let's see how it's passed through a family tree. Mutations and genetic diseases. So, mutations are changes to the sequence of the nitrogen bases along a DNA molecule. And this could be when there's been an error where cytosine has been added in the place of a guanine, or if a one has been inserted or deleted. So mutation is like a typo in a word processing document. So some might be small and make no difference at all. So like if we said we have 2.5 milliliters of salt or SAT, and we still understand what the recipe is asking for. Others might be more serious and change the whole meaning of the information. So 25 milliliters of salt instead of 2.5 milliliters of salt is going to make a huge difference into the final product. And the final product in our DNA is making proteins. So there's two different types of mutations. One is a point mutation. So this is when one nucleotide is substituted for another. So like I said, if a G has been inserted instead of a C. So instead of it saying the fox can run, this becomes the box can run. So the meaning of the phrase changed, but the individual words can still be understood. So this may or may not change the amino acid that is created when our DNA becomes a protein. Because remember, there's that redundancy. So we have like 6 or 4 or 2 different DNA triplets that will give us the same amino acid. Now, this could also be a frame shift mutation. This is when a nucleotide is either added or deleted. So adding a letter A to the phrase, dividing the letter into 3 codes changes every word. And now we're going to change every single amino acid down the chain from where the error was. So typically frame shift mutations are much more serious than point mutations when it comes to our DNA code. So we can see if we have our original DNA strand of ATG, GGA, GTT. If we change the first letter to a C, we get a new amino acid. We get leucine instead of methionine. However, and then the other two remain the same. However, in a frame shift mutation, if we deleted an A, we've changed all 3 amino acids. And if we've inserted a C, we've only changed 2 of the 3, but we still see that there's a bigger change in the amino acid change of the protein if we have those frame shift mutations. So mutations can occur because of an exposure to something in the environment that's known as a mutagen, like X-rays, which is why we wear those lead vests at the dentist, ultraviolet radiation from the sun, which is why we wear sunscreen, toxic chemicals, or other factors. A mutagen that causes cancer is known as a carcinogen. So something like the chemicals that have been added to tobacco smoke are known carcinogens. Mutations can get passed on to future generation. If they occur in your gametes. And we have some mutations that have occurred long enough ago that now the resulting condition is known as a genetic disease. And so this is a mutation on our DNA that affects our future generations. So if we have a mutation in one of our skin cells, this isn't going to affect the DNA of future generations. But if we have a mutation in one of our ovarian or testes cells, when sperm or eggs are being produced, this could cause the mutation to be passed on to future generations. So, an individual who possesses a disease copying mutated gene but does not develop symptoms is known as a carrier. So, we talked about this in the sex linked inheritance because women who have one normal vision chromosome and one colorblind chromosome are known as carriers. They have normal vision, but they have one abnormal allele. So, in this case, we're looking at sickle cell anemia. So if we have 2 parents who are carriers for sickle cell anemia, when we look at the alleles they're providing for their offspring, they have one normal allele and one sickle cell allele, and they both do. So when we look at their offspring, this individual will have 2 normal alleles, so they're not even a carrier. 2 of their 4 possible offspring's genetics are going to be carriers, and one individual could get both recessive traits, recessive alleles, and then be affected with sickle cell disease. So that carrier can pass on the disease-causing allele to his or her offspring. They could be carriers, or they could even be affected. If a disease is caused by a recessive allele, that individual requires 2 copies to be affected by that condition. So it's a homozygous recessive trait. If a disease is caused by a dominant allele, only one copy of the allele is needed for the disease to develop. And some diseases, like Huntington's, is autosomal dominant, where other diseases are autosomal recessive. So you need two copies of that gene to be affected. Then hemophilia is a sex-linked recessive trait because it's carried on the X chromosome. So males, if they have only one of the recessive alleles, will be affected, but females need two recessive alleles to be affected. So when we look at a pedigree chart, this is a family tree showing us the individuals that have a specific trait. So we have circles and squares representing gender. And then when the circles or squares are filled in, that means they're an affected individual. And an open circle will be an unaffected individual. So when we look at cystic fibrosis, this is an autosomal recessive condition. So that means that individual 1, so I1, has to have two recessive alleles. And so I'm just going to choose a lowercase f for fibrosis because it's easier to see than the capital F. And individuals that are not affected have at least one dominant allele. So when we look at individual 2, 2, we know that he has at least one dominant allele and one recessive allele from the parent who was affected. And so did their spouse because they were unaffected. But they also must have one recessive allele because they had a child that had the condition or two recessive alleles. So sometimes if you know that you're a carrier for conditions like cystic fibrosis or sickle cell anemia, you can get genetically tested to see if your partner's a carrier, to see if there would be any risk of having a child that's affected by that condition. So when we look at a pedigree chart for Huntington's disease, this is an autosomal dominant condition. So that means affected individuals only have to have one affected allele to have the condition. So it's much more likely that will be passed on. So that means that this individual has two unaffected alleles. Because when we look at the children, we can see that child 2, 4 and child 2, 6 and 8 are unaffected. We know that this individual has one affected and one unaffected individual allele. So, and again, that means that individual 2, 2, 2, 3, 2, 7, and 3, 3 all have that dominant allele, giving them Huntington's condition, Huntington's disease. So typically, if it's an autosomal dominant condition, you'll see it in every generation. Occasionally, there are some beneficial mutations, which then are going to allow the organism to have a better chance of survival. So if there's a black beetle that thrives on the surface of green leaves in the forest, if one individual has a mutation that gives it a greenish color, it's going to be more likely to survive and pass its genetics on to the future generation because it's less visible. For humans, one of the things that we developed as a mutation that was beneficial is the ability to digest lactose in milk because it gave us another food source. Now, beneficial mutations are part of natural selection, which is referred to as survival of the fittest. So the organisms most likely to survive because they are best suited to the environment are more likely to have offspring and pass on their genetics. So sometimes bacteria have a gene that makes them stronger and less likely to be killed by an antibiotic. So unfortunately then, when these bacteria divide, they are going to produce a population of bacteria that is resistant to antibiotics. And even though bacteria reproduce asexually, they're able to exchange their DNA between different bacterial cells to create more genetic diversity, resulting in new traits. And now this can happen very rapidly because bacteria reproduce very quickly. One of these processes is called transformation, and this is where free DNA is incorporated into a bacterial cell. The other method is by plasmids, where a self-replicating circular piece of DNA can be transferred between bacteria. So bacteria that have become resistant to antibiotics are able to share that gene and genetic material with other bacteria nearby. So antibiotic-resistant bacteria is quite a common problem in North America and other places around the world. So it's important to reduce our use of antibiotics unless it's necessary. I hope you enjoyed today's lesson. Catch you next time.